Welcome to the Deep Dive. You're listening because you want the key insights, fast. Today we're tackling two really important topics in epilepsy. First, the new 2025 seizure classification update.
That's right, hot off the press from the ILAE.
And second, we'll explore the complex world of developmental and epileptic encephalopathies, DEEs.
Yeah, we've looked at the latest ILAE guidelines and also a great webinar with expert discussion on DEEs.
So think of this as your shortcut. We're boiling down the essential info for you.
Exactly. We're giving you the core understanding, the why behind these changes and concepts.
Okay, let's jump right in with the seizure classification. The ILAE's 2025 update, it builds on the 2017 one.
It does. It builds on that foundation.
Right.
And this was a huge international effort.
Yeah.
You know, lots of experts, public feedback, quite thorough.
Sounds like it. So the big picture categories, have those changed?
Nope. The main classes are still there. Focal, generalized, unknown, meaning you're not sure if it's focal or generalized, and then unclassified. That basic structure works well.
Okay, familiar territory there.
Yeah.
So where are the refinements? What are the key things that are different in this 2025 version?
Well, one of the first things you might notice is they've actually dropped onset from the main names.
Oh, so instead of generalized onset.
It's just generalized seizure. Same for focal. It seems small, maybe, but it reflects evidence that some seizures look generalized right away, but might technically have a tiny focal start that spreads super fast.
Ah, okay. So it's acknowledging the practical reality of what we see, even if the absolute beginning is tricky. Interesting. What else?
They've also really clarified the difference between classifiers and descriptors.
Classifiers and descriptors. Okay, what's the distinction there?
Think of classifiers as the core biological features that really impact clinical decisions. So the main seizure class, focal, generalized, etc., the specific seizure type, and importantly, the level of consciousness. Those are classifiers.
Got it. And descriptors.
Descriptors are other important features that help describe the seizure, like what it looks like, the semiology, but they don't change that core classification for management necessarily.
Okay, so classifiers are the must-knows for treatment. Descriptors add the detail. Makes sense.
Exactly. And speaking of classifiers, level of consciousness is a big one. They've moved from using awareness to using consciousness for focal and unknown seizures.
Consciousness instead of awareness. Why that change?
Well, consciousness is seen as a more universally understood medical term and easier to translate accurately across languages, which is vital for a global system.
Okay.
And crucially, they define it operationally. It's not just subjective. Clinicians should assess both recall, which is like awareness and responsiveness, using specific tasks.
Right, so it's about actively checking if the person can remember and if they can respond, not just asking later if they were aware.
Precisely. It's a more objective clinical assessment of function during the event. Responsiveness can often be tested even when recall might be impaired or, say, in very young kids.
That sounds much more practical for actual clinical use.
Good point about translation, too. What about describing the seizure itself, the physical signs?
Yeah, that's changed too. The old split of motor versus non-motor for focal and unknown seizures is gone.
Okay, what's replaced it?
It's now a descriptor with observable manifestations versus without observable manifestations.
Observable manifestations. That sounds like more than just motor symptoms.
It is. It's any non-voluntary sign someone else could see. Motor activity, sure, but also things like, sudden difficulty speaking, autonomic changes like flushing or pallor, basically any outward sign. It acknowledges that even seizures we used to call non-motor often have something observable.
So it's less about muscles moving and more about any visible sign of the seizure happening.
Exactly. And they're also encouraging us to describe the whole sequence of these manifestations over time, the semiology as an expanded descriptor, not just the very first sign.
Right, because how it unfolds gives you more clues.
Definitely.
Any specific seizure types get recategorized. Yes, a few key ones. Epileptic negative myoclonus, that brief loss of muscle tone, is now its own generalized seizure type.
Okay.
Also, non-motor has gone from absent seizures because, well, they often do have subtle motor signs like eyelid fluttering or automatisms.
Sure.
And epileptic spasms are interesting. They remain a specific type of generalized seizure, especially think infantile epileptic spasm syndrome. But if spasms occur during what seems like a focal or unknown seizure, they're now listed as a descriptor, a semiological feature of that seizure.
Ah. So a spasm can be the main event in generalized seizures or a feature within a focal one. That's a useful distinction.
It is. And overall, the number of specific seizure types has actually been simplified down to 21 from, I think it was 63 back in 2017.
Wow, quite a reduction.
Yeah, the focus is on classifying based on all the info, the semiology, EDG, imaging, labs, genetics if you have it, and again, making sure the terms translate well.
It sounds much more integrated and practical. Could we run through a couple of quick examples how this looks in practice?
Sure. Let's say someone has a big tonic-clonic seizure, both sides involved, but nobody saw it start. Initial tests are inconclusive.
All right.
That would be classified as BTC bilateral tonic-clonic seizure, but with the note, unknown whether focal or generalized. It flags what we don't know yet.
Clear? And if you get more info later.
Right. Say the EEG then shows a clear starting point, a focus in the right parietal lobe, and the MRI shows some dysplasia there. Then it gets reclassified to FBTC focal to bilateral tonic-clonic seizure. The classification evolves.
Nice. How about a focal seizure example?
Okay, imagine someone hears something that isn't there an auditory aura. Then they can't understand speech, seem unresponsive, and are confused afterwards.
Okay.
That's FIC, focal impaired consciousness seizure. And you describe the sequence. Auditory aura, then receptive aphasia, impaired responsiveness, post-ictal confusion.
Wow.
That full description is key.
That detail really paints the picture, and a generalized one.
Maybe someone has quick little jerks, myoclonic jerks, right at the start, which then go into a full tonic-clonic seizure.
Right.
That's now GTC myoclonic tonic-clonic seizure. It specifically calls out that myoclonic beginning.
Highlighting important features. And those epileptic spasms.
So an infant having clusters of spasms with a typical generalized EEG pattern, that's GES, generalized epileptic spasm.
Okay.
But say an older child has spasms, but the EEG points to a focal issue. Maybe an MRI shows a lesion. That would be a focal seizure with epileptic spasms as a descriptor. See the difference?
Yes, the context and origin matter.
Yeah.
One more focal, but with consciousness preserved.
Sure. Someone gets that rising feeling in their stomach, starts chewing, fiddling with their hands, but they can still talk, answer questions, know what's happening. That's FPC, focal preserved consciousness seizure. And again, you list the observable bits. Epigastric aura, or elementary automatisms, gestural automatisms, preserved consciousness.
These examples really help. It feels like a very logical refinement, making things more precise and clinically useful.
I think that's exactly the goal. It respects the old framework, but incorporates what we've learned.
Okay, great overview of the classification updates. Let's switch gears now to developmental and epileptic encephalopathies, DEEs. This term seems increasingly important. What exactly is a DE?
Yeah, they're a really challenging group of conditions, mostly affecting children. Basically, you have two core problems happening together: significant neurodevelopmental delays or even regression, and epilepsy that's often severe and hard to treat.
So it's not just seizures happening alongside developmental issues, but they're somehow intertwined. Like the epilepsy itself might be worsening development.
That's often the concept, yes. And a huge piece of the puzzle we're uncovering is genetics. The webinar we reviewed mentioned, I think, over 900 genes now potentially linked to single-gene epilepsies. It's exploding.
Wow, 900 genes, that's a lot. With that genetic insight, what's the hope for diagnosis and treatment?
The big hope is precision medicine. If you can pinpoint the exact genetic cause early on, maybe you can find a treatment that targets that specific problem. And the idea is this could potentially help both the seizures and the developmental trajectory.
That would be a game changer. The webinar mentions some specific examples of conditions that can be DEEs, even with a single cause.
Absolutely. Things like CDKL5 deficiency or BCDH19-related epilepsy, those are single-gene DEs. But even some structural things like hypothalamic hamartomas causing those laughing seizures, gelastic seizures, can fit the DE picture because of the combined epilepsy and developmental impact.
So the underlying cause can vary, but it's the clinical picture, the severe epilepsy, plus the developmental issues that defines it as a DEE.
Exactly. And the webinar really dove into some specific types, starting with ion channelopathies. These seem really relevant for DEEs.
Ion channels.
Yeah.
They control the electrical firing of neurons, right? So problems there would likely cause major issues like seizures.
Precisely. Take KCNQ2-related DEE, often caused by loss-of-function variants in a potassium channel. Interestingly, sometimes high-dose sodium channel blockers can actually work quite well for the seizures in newborns.
Oh, well, that's counterintuitive, treating a potassium channel problem with a sodium channel drug.
It is, but it sometimes works for the seizures. The catch is the developmental problems often persist. There was an older drug, azogabine, a potassium channel opener, that showed some hints of helping both seizures and maybe development in a few cases, but it's off the market now. But it shows the potential, and new potassium channel openers are being developed.
So seizure control is crucial, but it's not always enough for the developmental side.
Hmm.
What about other channel problems, KCNT1?
Oh, yes, KCNT1, often gain of function, associated with really severe stuff like EIMFS, epilepsy of infancy with migrating focal seizures, and also a type of sleep-related epilepsy. These tend to be incredibly drug-resistant. Quinidine was tried, with some excitement initially, but results are pretty inconsistent. Seizure freedom is rare.
Drug resistance really is a recurring theme, isn't it?
It's a huge challenge, but there are some brighter spots, like KCNA2-related DEE. Gain-of-function variants here can cause significant delays, cerebellar issues, coordination problems. But a study showed that 4-aminopyridine, a potassium channel blocker this time, seemed to help quite a bit with seizures, cerebellar signs, cognition, even speech, across different ages.
Now that sounds promising, a targeted therapy based on the specific channel dysfunction showing broad benefits. What about calcium channels? CAC-NA1A was mentioned.
Right. Calcium channels are key for neuron communication. With CAC-NA1A, it gets complex. Gain-of-function tends to be a very severe DEE. Early onset status epilepticus, cerebellar signs, intellectual disability, episodes of weakness on one side, but usually no absent seizures.
Okay.
Loss of function in the same gene, though, often looks a bit different, maybe less severe overall, but with difficult absent seizures and moderate to severe intellectual disability. And frustratingly, standard calcium channel blockers or acetazolamide haven't been consistently helpful.
So the type of dysfunction in the same gene, overactive or underactive, really changes the clinical picture and maybe the treatment approach.
Exactly right. Function matters immensely. The webinar also hit on GABA receptor DEs. GABA is the main brake in the brain, the main inhibitory neurotransmitter.
Right.
You can have gain or loss of function here, too. One study found gain of function variants were linked to earlier onset, severe ID, focal seizures, drug resistance. And interestingly, they didn't respond well to GABA-enhancing drugs, which you might expect, but sometimes did better with sodium channel blockers.
Wow. Again, quite counterintuitive. Shows how complex the brain signaling is. And glutamate receptors, the gas pedal.
Yeah, glutamate's the main excitatory one. GRIN genes, for example, code for parts of the NMDA receptor important for learning. GRIN2A-related DEE is linked to speech problems. Variable ID, again, gain or loss of function matters.
So what are the approaches there?
For gain of function, memantine has had mixed results. There's a trial ongoing for a drug called ReadyPill, which dampens NMDA receptor activity. For loss of function, there's some early tentative evidence that maybe giving serine, which helps activate the NMDA receptor, could potentially help behavior, development, maybe even seizures in some cases.
It really seems like the field is trying to pinpoint the precise functional effect of each gene variant to guide therapy. The discussion then shifted to synaptopathies. That sounds broad problems at the synapse.
Very broad. The synapse is the communication junction between neurons, so synaptopathies can involve anything going wrong there: neurotransmitter release, reuptake, receptor, structural proteins. It's a huge category with diverse genetic causes leading to epilepsy, neurodevelopmental disorders, sometimes both.
Okay, and which specific synaptopathies were highlighted for DEEs?
One was SNAP25-related DEE. SNAP25 is involved in releasing neurotransmitters. This can show up as epilepsy with eyelid myoclonia-Jevons syndrome or myoclonic atonic seizures, often sensitive to light or eating. They mention a severe case, too, with intellectual disability, losing the ability to walk, reflex seizures, and even adult issues like sleep disorders.
So quite a spectrum there. What about SynJAP1?
SynTEP1 is postsynaptic on the receiving side. It helps regulate synaptic strength, crucial for learning. Loss of function here leads to too much excitatory signaling, not enough inhibitory. It's often linked to intellectual disability, autism, and frequently drug-resistant epilepsy. The webinar pointed out it's actually a pretty common single-gene cause for ID and autism with epilepsy.
That's important, recognizing the more common players and Shankopathies.
Right. Shank genes, Shank 1, 2, 3, also postsynaptic, involved in organizing glutamate receptors, linked to syndromic autism, often with intellectual disability and epilepsy, and sometimes that epilepsy can be particularly tricky to manage compared to epilepsy and autism generally.
So specific genetic pathways might predict a tougher course for the epilepsy. Okay, the last big category was mTORopathies. What's the mTOR pathway?
mTOR is a central signaling pathway inside cells. It regulates really fundamental stuff: cell growth, protein building, autophagy, which is like cellular cleanup. When this pathway gets abnormally activated in the developing brain, it can cause various malformations of cortical development, MCDs. And those MCDs often lead to epilepsy.
Malformation. So
Or
structural brain abnormalities present from early development.
Exactly. And there's a spectrum. Things like hemimegalencephaly, one whole side of the brain is too large, or MCK syndrome, tuberous sclerosis complex, TSC is a classic example, and also focal cortical dysplasia, FCD, where just a patch of cortex is disorganized.
And these are genetic too.
Yes, but it can be complicated. You can have germline mutations present in every cell, like in TSC usually, or somatic mutations, which happen after conception and are only in some cells or tissues. Genes like mTOR itself, TSC1, TSC2, PIK3CA, the GATOR1 complex genes, all involved. And when and where that mutation happens can affect how severe it is and whether it's focal or widespread. A later somatic mutation might just cause a small FCD, for instance.
That makes sense. The timing matters for development. Any clinical examples they shared?
Yeah, a case of hemimegalencephaly with seizures escalating to a really severe EEG pattern, needing hemisterectomy surgery. And of course, TSC, caused by TSC1 or TSC2 mutations. They talked about the typical brain findings, tubers, nodules, sagas, and the neuropathology within the tubers, like giant cells. They stressed how early epilepsy and developmental issues start in TSC, and showed a case where surgery on a specific tuber helped the seizures.
So even with these widespread genetic conditions, sometimes focal treatments like surgery are possible.
Sometimes, yes. The webinar also mentioned reviews looking at somatic mutations, finding mTOR mutations, often in FCD, PIK3CA, often in HMM-CAP, and also that germline GATOR1 mutations show up in a decent chunk of FCD cases too.
It's really painting a picture of a complex genetic landscape driving these developmental brain issues in epilepsy.
It is. And the conclusion from the webinar really emphasized that understanding these genotype-phenotype links is becoming so important. Even if genetic testing takes time or isn't available everywhere, knowing these patterns can help clinicians form hypotheses. But, and this was a key point, clinical skills, careful history, observation, they remain absolutely essential. You can't just rely on the gen test, especially with turnaround times.
So genetics offers powerful new tools but doesn't replace good clinical medicine.
Exactly. They work together.
Well, this has been incredibly informative, covering both the nuances in the new seizure classification and the really complex, evolving landscape of DEEs. If you had to boil it down, what are the absolute key takeaways for our listeners?
Okay, first, the 2025 ILAE seizure classification. It's a refinement, making the system more practical, more precise, based on current evidence, but keeping the core structure we know. It improves how we categorize and communicate about seizures.
Right.
Second, DEEs. These are tough conditions involving both severe epilepsy and developmental impairment. The genetic understanding is just exploding, which is incredibly exciting because it opens doors for targeted therapies that could potentially impact both seizures and development down the line.
It definitely feels like a field in rapid motion. Research is constantly shedding light on what these gene changes actually do. So as we wrap up, here's something for you to think about. With genetics and precision therapies advancing so quickly, what might epilepsy diagnosis and treatment look like for people with DEEs in, say, the next 10 years? And how will this updated, more precise seizure classification help us measure progress and tailor those future treatments?
That's the big question, isn't it?
Definitely food for thought. We really encourage you to check out the ILAE resources and keep an eye on the DEE research. Thanks for joining us for this deep dive.